Okay. So this was the concept that we talked about in terms of the neuronal action potential, right? Resting membrane potential, the sodium ion channels, ligand gated versus voltage gated. Yes, does that ring a bell? So all of these things that we talked about. Now, I just want you to see this slide real quick. This was something that we talked about. Remember the myosin head attached to actin, that is the contraction, yes or no? Right? So this is what we are trying to achieve. Now, this ATP coming in, what does it do? The first thing that it does is it detaches the myosin from actin, yes or no? Right? And then that ATP undergoes hydrolysis, correct? Now, who can tell me what should the myosin head have in order to do this hydrolysis process? Again, remember, the assumption here is we know some intro physiology, yes or no, right? So please make sure to kind of brush up on the background and see if you can remember what is in the myosin head that helps do the hydrolysis. Anyone? Remember, for hydrolysis, what do you need? You need water and an enzyme, correct? Okay, so the enzyme would be ATPase. So the myosin has an inbuilt ATPase enzyme inside of it. Okay, as soon as it detaches the ATP, what does it undergo? It undergoes hydrolysis. Got it? Because the myosin head has the ATPase enzyme inbuilt into it. I think that's an amazing thing. Very tiny myosin in all of our 600 odd skeletal muscles. Imagine the amount of myosin head and the ATPase inbuilt. Okay. The reason why I pointed out this is, now look at this. Have you heard of rigor mortis condition? Yes. So what is the first thing that happens when ATP comes in? ATP comes in, the first thing is it detaches myosin from actin, correct? Now, if ATP is not there, what do you think will happen? The myosin will be attached to actin all the time. Yes or no? All right, and that will cause the muscles to become stiff. The stiffness of the muscles that occurs following death is what we call rigor mortis. You guys with me? Yes, this is because of lack of ATP. All right, again, if something is not clear, please book an appointment with my office hours or stop by during tutoring or any other time that you are available. Please make sure to schedule something with me so that I can give more clarifications. Okay, so please avail those opportunities. I'm here for you all the time. It's just that we have to make sure that we have to match our schedules. That's all. Okay. So motor unit, as I told you earlier, the skeletal muscles bank on what? Neuronal stimulation. Yes or no? Right? Without the neurons, the skeletal muscles do not work. Okay, we call that as a combination of motor unit. Motor unit is a combination of somatic motor neuron. The term somatic is used to address skin and skeletal muscles. Okay, so that's why you use the term somatic. What is the another term that we use for cardiac and smooth? Anyone? Somatic nervous system. The other one would be if they're operating on their own. What do we call that?
Autonomic.
Thanks, Malak. Yes, autonomic nervous system, right? That would serve the cardiac and smooth muscles, okay? So motor unit is a combination of somatic motor neuron plus all the muscle fibers that it innervates, okay? So if there is no innervation of motor neurons, the skeletal muscles will also not. Got it? Right? And this is different types of innervation, different ways of how, because some muscles are part of fine motor skills, right? Some muscles are made for major work, okay? So types of skeletal muscles, this is where we bring in a little bit of biochemistry here. So I want you to focus on one. The other one would be the opposite, correct? So we will just focus on one first. Now here, The types of muscles are categorized based on fast versus slow. So we're going to talk about slow first. So slow, obviously the process is slow. And then they undergo oxidative phosphorylation. Does that ring a bell? Oxidative phosphorylation, Krebs cycle, mitochondria, electron transport chain, right? I'm sure you have heard of all these, right? So this is where the oxygen is required. So why do you think we talk about oxidative phosphorylation, oxygen requirement? What does this have to do? What is the connection that I'm looking for? We just talked about that. Something is required. Yes, Malak. Very good. Thanks, Malak. ATP, right? So oxidative phosphorylation gives me the ATP that is required for skeletal muscle contraction. How the muscles get ATP is how we divide these fibers into. Make sense? Right? So slow oxidative type 1 fibers get it from oxidative phosphorylation. It seems that they have a smaller diameter, okay? And then high, I have something on the chat. Let me quickly see if I need to address it. All right, thanks, Christy. So high myoglobin content. What is myoglobin? Very similar to what you see on the red blood cells. What do we have? Hemoglobin, right? So myoglobin has a lot of oxygen content. Remember, oxygen is required. High capillary. Capillary is a blood vessel. What does blood vessel bring? Come on, try to engage, please. It's fun to just kind of talk.
Oxygen.
Very good. Yes, blood brings oxygen, which means there's a lot of capillary density. Capillary here is blood vessel. Okay. I see half the students have turned on the cameras. I'm still waiting. Right. Many mitochondria. Thank you for this. Mitochondria, why is mitochondria? What is the relation here with this process? Yes or no? Mitochondria is where oxidative phosphorylation happens. And then if everything is oxidative, what do you think will happen to glycolysis process? Less, right? So low glycolytic enzyme content. Got it? Okay. So everything is in favor of oxidative phosphorylation. So the slow muscle, they're also called red muscle because there is more blood vessels bringing in more oxygen, right? Myoglobin, what gives the color red to the blood? The heme portion, yes or no? Right? Very similar to, we have myoglobin. So it's called red meat. Have you heard of people saying red meat, red muscle? So what is the connection? Red muscle, oxidative phosphorylation, slow oxidative fibers. Are you guys with me? Right? So if you know this, what would this be? The opposite. Right? So don't try to remember both. Confuse yourselves and me. Right? This is going to be fast fibers type 2. What is it predominantly? Glycolysis, correct? If that was small, this is large diameter. If that was high myoglobin, this would be low myoglobin, correct? If that was high capillary, low capillary. Many mitochondria, few mitochondria, low glycolytic, high glycolytic. How easy is physiology, guys? You can do this, right? We can all do this. Got it? Okay, so slow red muscle, and because this is devoid of a lot of myoglobin stores, how would the color be? White. Got it? So white muscle versus red muscle. It would help if you can remember the alternative names. Slow fibers type 1 oxidative, fast fibers type 2 glycolytic. Sometimes some individuals can have a mix of both, yes or no? Somebody like me, I want to do exercise one day. I don't want to do exercise one day. So I might harbor both glycolytic and oxidative energy, right? But that's something that's dependent on the individuals. Any questions? Are we good? Yes. What is an important element though? How do you link that to something that we can see on a, not on a daily basis, but on our like daily lives in terms of who do you think would harbor red muscle in terms of athletes? Like what kind of athletes would
Marathon runners.
Very good. Thanks, Laura. Because they are in constant supply, they get constant supply of oxygen. Yes, right? So marathon runners would harbor oxygenated fibers. But somebody who is a sprinter, right? They need quick bursts of energy, which means they burn glucose, anaerobic, correct? What is the difference though? Marathon runners, they get constant supply of oxygen, so they are resistant to fatigue. They don't get fatigued, right? Whereas sprinters, yes, they get quick ATP, not very efficient, but they get it, but they fatigue easily. Got it? So those are the application-based differences based on these two fibers. Everybody good? Yes? Moving on.
Professor,
Yes.
I have a question about a previous slide. I don't think it's on your PowerPoint.
Yeah.
The one on skeletal muscle length tension, the two graphs you have on 46 and 47.
Okay.
Were you planning on going over that?
I can go over it later. Yeah.
Okay. Okay, thank
Okay.
you.
I can bring that up after I'm done with this. Yeah. I mean,
Okay,
I'm
thank
trying
you.
to focus on the major ones, but you can always go over that on my recording. And if you have any questions, you can put it on the chat. I can look it over the break time and bring it up after the break. Is that okay?
Okay, thank you.
Yeah. All right, thanks. So neuromuscular junction, remember, we are slowly going from being in depth to slowly going upstream, yes or no? So we know what happens in the myosin actin level. We know the importance of ATP. We know the importance of calcium. Now remember, what do we need? We need a motor neuron to cause something at the skeletal muscle level. So what is this called? Neuromuscular junction, okay? Remember, I'm not going over every slide, guys, as part of this, because we want to make sure we are able to cover the entire concept, okay? So again, I'm willing to go over any slide that in particular, like this, like Malik suggested, after I'm done with the concepts, okay? All right, so please feel free to bring that up. So again, these are some of the terms that they use for synapse. So this is a synapse between a motor neuron and muscle. So neuromuscular junction. What is crucial here? You should know the steps. So first, there is action potential happening, and it reaches the axon terminal. Then the calcium channels, they are voltage gated, they open, the calcium goes in, and that prompts the release of what important neurotransmitter? ACH. Yes or no? So ACH gets released, and they look for ACH receptors. Yes or no? Where are those receptors? They are on the skeletal muscle, waiting to bind with ACH. And upon binding, remember the acetylcholine receptors, they can also act as ion channels. Yes or no? Right? So can anyone tell me what kind of ion channels am I looking for? So I am the ACH receptor on the skeletal muscle. ACH is binding to me, and I'm slowly releasing sodium ions, trying to bring the membrane potential close to threshold. What channel am I?
The one on the post, like on the muscle?
Yes. I'm the receptor. I'm the ACH receptor. I'm behaving as a channel. So upon ACH binding to me, I am releasing sodium ions slowly. Yes, Mala?
You're saying which one bind? So acetylcholine would bind to the nicotinic
Very
acetylcholine
good.
receptor?
Yes, nicotinic ACH receptor. You see that ACH receptor? I want you to just put N for now. Nicotinic ACH receptor, which also behaves as what?
A ligand gated.
Yes, go. Sorry. Ligand gated. Yeah. Sodium channel. Got it? What is the importance of ligand gated sodium channel? What is it? This is trying to bring the membrane potential close to anyone? Remember, sodium is slowly coming in, right? The muscle is at rest before. Now sodium slowly coming in, will get the membrane potential close to threshold.
Threshold.
Yeah, correct. Thanks, Lauren. And this will, you see this? Sodium entry, right? This will cause the opening up of voltage-gated sodium channels. Why is this needed? This is for what? Voltage-gated sodium channels. Immediately, you should connect that to what? It's right there. Come on. Yes.
Action potential.
Action potential. Voltage-gated sodium channels. This should come in your dream. Voltage-gated sodium channel linked to action potential. Yes or no? Okay. And action potential is what stimulates the membrane. Correct? All right. And that will help bring in what ion inside the cell? What is it? An ion that is needed for skeletal muscle contraction that helps tropomyosin and troponin, all of the fun stuff that needs to do. Any takers on what ion?
Calcium.
Calcium. Thank you. Thank you, Trevor and Malak. Yes, very good. Got it? So voltage-gated sodium channel. Are we clear? And another important thing is, as soon as ACH binds to the ACH receptor, it's like a relay, right? It relays information. So ACH has to be taken off from the junction. And what comes to do that? ACHE, acetylcholinesterase. So this is an enzyme that helps remove or take away the ACH. Got it? Okay. Now, again, remember, all of these are great application-based questions that could be asked on the exam. If there is a drug that stimulates too much of ACHE in the body, what is it trying to do? If there is a medication that causes overactivation of acetylcholinesterase in the body, what do you think it is doing?
There's going to be less contraction.
Less
Because less is
ACH,
happening.
yes, less ACH available. Yes or no? That esterase enzyme is going to eat away all the ACH that's being released. There's not going to be ACH available for contraction. But this could help to someone who is releasing more ACH than needed. Yes or no? Right? If somebody is making too much ACH in order to control their symptoms related to too much contraction, this medication can help. Right? Remember, medications are always going to be the way we use it for. Okay? It could be detrimental. It could be beneficial depending on what and how we use it. Got it, everybody? All right. So please remember when you study, hey, this is what this enzyme does. This is what is the process. What if it goes wrong? What if it gets overstimulated? Or what if it is underactivated? Also keep that in mind when you're studying. That really helps with application-based questions because that is what advanced physiology is all about. Yes or no? These information are actually from introphysiology, right? I'm just kind of reviewing this, but how do you put it in use is most important, okay? Again, just to kind of bring this up, knowing this process, right? So it is important that you know all the steps involved. Now look at this. What is this drug doing? It is blocking nicotinic ACH channels. What is it trying to do? Just don't even look at it. What will this do? You guys are used to multiple choice, right? Contract, relax. Which one? It is blocking nicotinic ACH receptor. What is happening to the muscle? Come on.
It's going to be relaxed.
Thank you. Very good. Thanks, Lauren. Right. Because nicotinic ACH receptors is not going to get activated. No muscle contraction, right? That is why D-tubochlorine is a muscle relaxant. We all the time come across muscle relaxant that's used in surgery, yes or no? Right? So this is one of the drugs that are being used. So why? Because we don't have activation, there is no action potential eventually happen. So no contraction. Now, how many of you have heard of Botox? right? So Botox is also going to help with relaxing the muscle, yes or no? But the mechanism is different. What is the mechanism? The mechanism is to decrease the release of ACH from nerve terminals. Okay, so here, remember, the ACH is released, correct? So the Botox basically acts on this area and prevents it from releasing it. Okay, there is, again, an hour lecture just goes into how this mechanism works, but I'm just giving you an overview here. Every tiny bit of information that we look upon, right, in physiology has by itself an ocean of information. All right, so I'm just trying my best to keep it simple, but at the same time touch upon few things. Are we good? Right, so no ACH released, no NACHR, no threshold, no AP, nothing's happening. Got it? So it's a muscle relaxant. Got it? Both are muscle relaxants. What is important? The mechanism of action, how they are working in the body is what's different. Okay. And of course, they have their own ways of giving us, because every medication has its own side effects. So that's why many different processes are important to understand. So myasthenia gravis, just to link a clinical condition to this. So this is where you see sometimes lethal paralysis can happen. The cause is autoimmune disease. So why this happens is because our own autoantibodies destroy the nicotinic ACH receptor. Remember, nicotinic ACH receptor is the receptor that is required for skeletal muscle contraction, yes or no? Right? If it's destroyed, then obviously we are going to have weak potentials. So basically less stimulation, less contraction. So this is a popular medication, histamine. This is anti-ACHE. What does that tell you? So ACHE is acetylcholinesterase. Yes or no? That is the abbreviations. Abbreviations are important. If it is anti-ACHE, so what does this do to ACH? What does this do to ACH?
It blocks the breakdown of ACH, which is what ACHE does, so that there's more ACH available.
You are correct if you had talked about anti-ACHE. I'm asking about ACHE. This is going to?
Gotcha. ACHE degrades the ACH in the muscular
Perfect. Thanks,
junction.
Laura. Yes. Your answer previously was for?
The anti-ACHE.
Exactly. Thank you very much. Yes. Which means this is going to increase ACH. It seems that the symptoms improve by doing this. So they are bombarded with a lot of ACH, and it seems that new receptors populate which are not destroyed, and that helps with kind of symptoms of the myasthenia gravis. Again, there's a lot of hypothesis, but this is working, so we just okay, which increases the ACH. Got it? Okay. So again, we can prescribe steroids to suppress the immune system. Yes or no? Prednisone is a steroid to suppress the immune system, and plasmapheresis to remove antibodies that are destroyed. So these are some of the techniques. Are we good? Myasthenia gravis. Okay. So this is the overall. Now everybody understood this ATP requirement, thick filament, thin filament. Everybody good with this, right? Myosin actin binding. What do we need? Calcium. Now our point is, yes, the muscle is stimulated by a motor neuron, correct? And for this, what do we need? ACH, NACHR, all those, correct? And then an action potential is generated. This action potential has to actually travel inside. This is where we love anatomy. Who loves anatomy? I do. Right? So these transverse tubules that you find here will take the action potential deep inside the muscle, where we have these cute little channels, DHP and ryanodine. So these receptors, the DHP is on the muscle, ryanodine is on an important organelle called sarcoplasmic reticulum. The name sounds similar to endoplasmic reticulum, yes or no? Have you heard of endoplasmic reticulum? Smooth ER, rough ER, 1200 biology. Come on, right? So ER. Here we say sarcoplasmic because of the sarcolemma that is kind of unique to skeletal muscle. The sarco is a prefix that we add for anything related to skeletal muscle. Got it? So sarcoplasmic reticulum, why is that organelle important? Because it has a large amount of calcium in it. Okay, so this calcium is inside and it's released. Then in the calcium, obviously we know that calcium goes and binds with troponin, moves the tropomyosin away, allows myosin to bind with actin, provided the myosin is energized, blah, 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 all of those fun stuff that we already covered. Okay. Now you see the importance of these channels, guys. DHP receptor is getting the action potential information and delivering it to a ryanodine receptor. Okay. And now, how many of you have heard of malignant hyperthermia? Have you heard of that condition at all? Or look it up later. Malignant hyperthermia is one of the major things that can happen during surgery. Okay. Because of skeletal muscle, because there's a lot of heat generated during the surgery. Sometimes the body temperature can shoot up, right? And that can also cause hyperthermic condition. And that's usually triggered. Typically, they try to relax the muscle, right? But some anesthetic given to the patient can trigger this ryanodine receptor and overactivate it. So that's one of the detrimental use of using anesthetic. So genetically, they might be susceptible to it, and this anesthetic can trigger that overactivation of ryanodine receptor. What happens if there is activation of ryanodine receptor? There's going to be more calcium pouring in, yes or no, which will cause the skeletal muscle contraction to keep going on and on. What happens if when skeletal muscles keep contracting? They generate a lot of heat. And then the body temperature shoots up. And that is not. Got it? So this is malignant hyperthermia. And now another important thing that in terms of mechanism that I want you to understand is, you see this calcium, guys? It comes from SR. Yes or no? There's a lot of calcium here. And it comes from SR into the sarcoplasm. Yes or no? Which is the cytoplasm of the skeletal muscle cell. Now, this calcium that came in has to go back to SR when the skeletal muscle relaxes. Yes or no? Right? So if it relaxes, I don't want calcium in there. I need to get the calcium back to where it belonged to. Now, it is going from relatively going to its home, right? So this is kind of low to high. Yes or no? That's where it came from. It's going to go back. Just like how sodium will be kicked out to where it belongs to, calcium needs to go back, which means what does it require?
Active transport.
Very good. Pump. So what is the pump that we are looking here? Calcium ATPase pump. Yes or no? Remember, ATPase hydrolyzes ATP to give the energy for the pump, to pump calcium from the sarcoplasm back into the sarcoplasmic reticulum to cause relaxation. Beautiful, right? So what can we conclude? ATP is required for both skeletal muscle contraction and relaxation. That is why they say, hey, you're doing exercise, take a break, take a break. Intermittent, right? HIIT. I'm not familiar with those abbreviations, guys. So, but that's those interval training that they say, right? Take a break between exercise. Why is that? Even when you're relaxed, what are you still burning? ATP. Yes or no? Right? Because ATP is required to pump it back, get it back, pump it back, right? It's amazing how we use our physiology for a lot of things in our daily lives. Are we good? Yes. Okay. So skeletal muscle, cardiac muscle. I want to start the next set of chapters. So skeletal muscle and cardiac muscle, very similar in terms of how they look. Of course, cardiac muscle is involuntary. Yes or no? Right? And remember, any muscle requires what? What is the first step? Stimulation. There's no doubt about it. Skeletal muscle, they have a dedicated motor neuron. Okay? Whereas cardiac muscle, even if I take the heart from my body, it can still beat. Listen, because they have those inherent pacemaker. Correct? So these pacemaker cells, they have the capacity to initiate stimulation on its own, right? But stimulation is still there, whether you take cardiac or skeletal. Got it? Okay. Involuntary. What do you think will happen to nervous system in the case of cardiac muscle? Are they going to still stimulate cardiac muscles? Do they have sympathetic, parasympathetic activity on cardiac muscle? Yes or no? Right? So remember, the heart can beat on its own because of pacemaker cells. But the sympathetic innervation makes it beat faster. Parasympathetic makes it go slow. So the nervous system still can regulate how frequently they can beat, but they can still beat on their own with the difference, basic difference between cardiac and skeletal, because skeletal muscles, they do not have pacemakers. Make sense, guys? Okay, all right. Now, the assumption is they are stimulated. Both of them are stimulated. Now, they still need calcium. Calcium is a big part in both cardiac and skeletal. The major difference here is the calcium is entirely released from the sarcoplasmic reticula. Right? Okay, as soon as the action potential is there, calcium. But here, the calcium is released from outside. We call that ECF, as well as from S. So there is a calcium channel that's found on the muscle cell membrane of the cardiac muscle that allows calcium to come from outside. Then it goes to the SR and say, hey, get more calcium. Okay, so that's a basic difference in how the calcium is handled. You still have the DHP receptor, and then you still have the ryanodine, but the DHP receptor is actually a calcium channel. Okay, unlike what you see here. The DHP here is only sensing action potential. Listen. All right, and this action potential is conveyed to the ryanodine receptor. Here, the calcium is actually telling, hey, ryanodine receptor, get me more calcium. So calcium is talking to the ryanodine, so it's calcium activated calcium release. How beautiful is that? Right, calcium activated. But here it is voltage activated calcium release. The action potential is telling ryanodine to release. Got it? As much as possible, apply logic. There's a lot of logic behind these physiological mechanisms. If you put your mind to understand the logic, it stays in your mind, and you would trust me, you can become teachers. Okay, so just put it in your mind the way you understand. Okay, and that's why I love physiology. Right? If it doesn't make sense to me, I try to dig in until it makes sense to me. Okay, are we good so far? Yes. Okay, that's a basic difference. But other than that, myosin, actin, ATP, everything is the same. How beautiful, right? Except for these changes that I told you. Pacemaker, involuntary, calcium. Can you remember these basic differences? Okay, all right. Obviously, cardiac muscle is going to be on the heart. That's another difference, okay? All right. This is smooth muscle. Again, a very broad understanding, but some nuances to it. Smooth muscle is smooth, so they don't have that pattern, no sarcomere, so no sarco prefix. Typically, they should be replacing this with endoplasmic, right? Because there's no sarco here. Smooth, okay? What do you see here? Smooth muscle. Can you compare that with cardiac and skeletal and tell me what is incomparable to skeletal? What is incomparable to cardiac? It's comparable to cardiac in terms of involuntary, correct? And it has pacemaker. At least most of them have pacemakers. Got it? and also calcium activated calcium release. Yes or no? Similar to cardiac, correct? So these are all similarities. Again, remember, similarities are much easier to categorize and remember, okay? What's the difference though? So skeletal muscle obviously has myosin, correct? Myosin actin. Here, there is no troponin. So when you compare it with skeletal muscle has troponin. Here, there is no troponin. It is replaced by calmodulin. So calcium is still an important ion. It binds with calmodulin and not troponin. That's the big difference. And then the myosin, we don't call it myosin head. We call this myosin head more in skeletal muscle. Here, this head, we call that as light chain kinase. So we call that as myosin light chains. That's all the differences. It's the same thing. It looks like head. We just don't call it. We call that as myosin light chain. Now, the big difference here is how are they energized? Just like how we saw ATP involvement, correct? The ATP is also involved here. The difference here is this myosin light chain will be adding a phosphate. You see that? So you add a phosphate to it, it becomes a phosphorylated myosin. It is now energized and ready to bind with actin, causing contraction. Got it? Yes? So any biochemistry, what enzyme helps with phosphorylation? I already told you. Yes.
Kinases.
Kinase. You see that K? So MLC is myosin light chain. If I add a K, it becomes an enzyme. So kinases add phosphate to any molecule that it is involved with. So if I have MLCK, then it will phosphorylate MLC. Yes or no? Right? If I have AK, it is going to phosphorylate A. That's it. So this is contraction. What do you think will happen for relaxation? You need to take away those phosphate, right? So phosphatase comes in. This is the opposite of what? Opposite of kinase. It takes it, then the calcium, do you think has to go back, right? Yes or no? So calcium has to go back. Again, it requires ATP, and then everything is back to normal, right? Contraction versus relaxation. So I tried my best to give you a glimpse, at the same time, a little bit more information on the details about different things that you need to go over for nerve and muscle. Please remember to match up anything that I talk about during synchronous as well as asynchronous material to the study guide. Study guide is your go-to document that you need to add. Keep adding, reviewing, clarification. I'm here to help. Are we clear? Yes? I'm going to have some review questions. So we are going into the nervous system A and B chapters. So this is where we touch upon a little bit of sensory physiology and some general principles. Again, this is some of the basic foundations that kind of leads us into the information related or concepts related to sensory receptors, okay? Again, as I told you, please be patient as some information that I want you to explore and try to be aware of these topics, but we are not every topic is deep dived into, okay? So please make sure that you are aware of the pace of the topics covered. So sensory division, integrative division, motor division. So we have, this is the, so we have the central nervous system, the brain and the spinal cord, correct? Right. This receives input from the afferent. What is afferent also called?
The sensory?
Yes, the sensory. Okay. The sensory information is received from somatic senses, skin, skeletal muscle, special senses, the eyes, the ear, right? In the visceral senses, even from the internal organs, the sensory information can be received by the central nervous system for process. Okay. And then the processing happens here and it's delivered output to efferent. So sensory division, then integrative division, processing information, motor. So efferent is also called motor. Okay. And this motor division goes to somatic. Yes or no? We just saw the ACH involvement, correct? ACH, ACHR. Then autonomic, sympathetic. We will touch upon this in the nervous system C set of chapters, sympathetic and parasympathetic. And that is autonomic because we have these muscles that can operate on their own, correct? Right? So cardiac muscle, smooth muscle, glands. And there's also a dedicated nervous system called enteric nervous system that is for the GI, gastrointestinal tract. Okay, so these are all the major organization of the nervous system. Any questions for me on the major divisions? You good? All right. Now, just to see the function of dendrites. Again, dendrites are the ones that are receiving the information. Okay. So dendrites allow signal reception and they do not transmit action potentials. So which means they typically are, they are devoid of voltage gated sodium channels. They have only ligand gated channels. Yes or no? Right? Because their job is to bring it either close to threshold or away from threshold, depending on whether it's the excitatory or inhibitory, correct? We talked about that. So remember, these kind of potentials that are generated by ligand gated are called graded, correct? Graded potential. What are the other names for graded potential? Electrotonic potential, local potential, end plate potential. I'm sorry. There are different names. Synaptic potential, receptor potential. They're all different names. But those graded potential is definitely not an action potential. Correct? Right? Synaptic, end plate. So end plate you will see on the muscles. Synaptic you will see on the neurons, right? Receptor you will see on the sensory receptors. So they just pick the names based on where you see. Got it? But these are all graded potential. Any questions? Good. So signal transduction at synapses. Now here you see this is a receptor. The receptor is behaving as an ion channel. Yes or no? Right? So what is a classic example that we just saw in skeletal muscle? A receptor behaving as an ion channel. Can anyone give me a quick name to this? An example?
The nicotinic acetylcholine receptor.
Very good. Thank you very much. Nicotinic ACH receptor is an example of a receptor being a ligand-gated channel, right? So if the receptor is in the form of a channel, we give the name ionotropic. Got it? That's important. So this receptor is behaving like a channel, but it also has an opening for what? A ligand, correct? So ionotropic can also be called as ligand-gated ion channel. Got it? Okay. And again, what are we looking at? Graded potential. Yes or no? If this is a potassium ligand-gated channel, ligand-gated potassium, what is it trying to do to the membrane potential? Make my day here. If this is not this one, so NaCHR is actually ligand-gated sodium ion. This is going to bring in sodium, yes or no? Causing the membrane potential go close to threshold. So if it is another example, if it is a ligand-gated sodium channel, what will happen to the membrane potential? Come on, you can do this. You know where potassium is high.
It'll decrease.
Yes, it will go away from threshold. Yes. Yes or no? Because it will cause potassium ions to go out. Right? Thus, the cell will decrease the amount of cations, bringing it more negative. More negative means away from threshold. Do you see how we built on our knowledge, right? Okay. Now, what is this? Are you familiar with G protein receptors from your earlier classes? So G protein receptors, any receptor that's coupled to a G protein, we give the name metabotropic. So metabotropic is a term given to a receptor that's coupled to G proteins. Got it? So the receptor is not an ion channel. It is coupled to G proteins, which has alpha, beta, gamma. Again, we are not going into the mechanisms of all of this. This is by itself is a separate 2, 3, 4 hour lecture. But activation of G proteins will eventually cause a second messenger. Eventually, what is our goal? Our goal is still an ion channel, yes or no? Right? Whether it's a direct one like this, inotropic, or here it's eventually trying to either open or close. But what is it trying to do? It gets the help of a G protein coupled receptor. And we have several of them, right? G protein coupled receptor. Got it? So I want you to keep in mind, metabotropic receptors are receptors that are coupled to G protein, and there could be receptors of this type that eventually helps open or close ion channels. Got it? Okay. Which will still affect what? What is our goal? Opening or closing will still affect membrane potential. Yes or no? Right? That is our goal. Okay. Now look at this, ACH receptors. Tell me something that you already know. Do we know nicotinic ACH receptor? Yes. Do we know that they are found in neuromuscular junctions? Yes. Okay. We haven't talked about ganglia and stuff like that, so just focus on the ones that we know for now, but we can always integrate. Okay. And curare, the D-tubocurare medication that we just talked about. Yes. Curare, muscle relaxant. Does that ring a bell? Right. So this is something that curare blocks what? And ACH. Right. That's the relaxant, muscle relaxant. So we have ACH. So this is NACHR. Okay. So there's more sodium coming in. So this is the ligand-gated sodium channel. Ignore these K plus for now, because it's going into the intricate channel concept. So this is ligand-gated predominantly sodium channel that will help with depolarization, action potential, causing excitation. Got it? So what is the point that I'm trying to make? Every time NACHR is activated, there is always going to be excitation. Got it? So NACHR, when activated, is always going to be causing excitation. So we will see NACHR also in the brain. Right? And when this gets stimulated by something that we are pretty familiar, nicotine, yes or no? Nicotine addiction. Do you see what I'm trying to say here? So when somebody is getting addicted to nicotine, what are they actually activating on and on in the brain that makes them excited, not because you're learning physiology. That's a different story. Nicotine, right? So that's why it gets the name nicotinic ACH receptor, because that activation by nicotine causes excitation all the time. Isn't that amazing, right? How we kind of reduce those things. Another class of ACH receptor is muscarinic MACHR. Okay, so they are found on multiple regions in the body. So they are not, so this is nicotinic is ionotropic. Muscarinic is metabotropic, which means they are coupled to G protein. So, see that? Right. So this is an example. So example here would be cardiac muscle on the left. And this is GI. Again, we don't have to go into the nuances, but you will see that this is a G protein coupled receptor, correct? Binding with ACH, right? What is the outcome? Can it be always excitation for a muscarinic receptor? It can be inhibition or excitation, mixed response. Okay, so when muscarinic receptors are activated, based on where they are located, the response is different. That's what I'm trying to say when compared to NACH. Got it? Yes, for now, let's leave it like that. When we talk about parasympathetic, it will make much more sense. Are we clear? The difference between nicotinic versus muscarinic. And again, we will come across this later, but I don't want to bring in atropine right now. But atropine, if you want to just kind of compare it with curare, if curare is blocking NACHR, what do you think atropine does? It would block MACH. Remember that? No? It's a blocker for MACH. Got it? Just to compare the left versus the right receptors. Any questions? So what factors generate receptor potential? So here, as you can know, receptor here, we are moving on to the sensory portion here. The sensory receptors, they all are triggered by changes, yes or no? If you're hearing me talking, which means your hearing receptors are changing its potential, yes or no? Based on my sound waves coming to your ears. If you're seeing something, your eye receptors are getting activated, correct? So they are responding to changes, mechanical, chemical, temperature, electromagnetic, yes or no? Every time they see these, what are these called? External stimulus, yes or no? If they're coming from outside the body. Every stimulus is trying to cause a change in the membrane, correct? Okay, this membrane is going to either make it close to threshold or away from threshold, correct? And that will cause AP or not. So the process is the same thing. Remember, all of these external stimuli will lead to change in the permeability of the ions. It can either cause hyperpolarization or depolarization. What does that mean? It can either bring it close to threshold or away from threshold. Got it? If I'm not talking, no threshold. If I'm talking, threshold, as simple as. Get it? Right? And that is how, what we call the term signal transduction. Remember, our body only talks in terms of what? Action potential, right? Every stimulus has to be converted to voltage. That's how it reads it. That's why we use the term firing of neurons. Have you heard of that term? The neurons are firing every time a stimulus is being sensed. Oh, beautiful, right? And we know this. Any receptor potential, who can make my day? What am I looking for? This starts with a G. Any receptors, initial activation will always give rise to
Graded potentials.
Thank you very much. Right? Why no action potential except in the axon? Definitely you can go for a break after this answer. Why no AP on the receptor cell here, no AP here. Only the AP is on the axon.
Because of the sodium channels?
What kind? The close. What channels? What
The
sodium?
voltage, the
Yes.
voltage gated.
The voltage gated sodium channels are found only on the axon and not on the receptor. Beautiful, right? We'll take a short 5-minute break. Then I will probably see everybody back, right? Have a good break, guys. No worries. So based on what we just saw, chemicals, temperature, right, mechanical, so we can classify the somatic sensation as mechanoreceptive. Yes or no, right? Mechanical. It responds to mechanical changes. So one example could be touch. And this is proprioceptive is position. And do you know of any mechanoreceptors that could be present inside an internal organ? Can you kind of out of the box thinking? Mechanoreceptors that can
I think the ear,
monitor. Okay. Yes.
the ear, because of the vibrations.
Okay. That is still sensing the external stimulus, but some internal stimulus.
Hmm.
I'm going to give you an important vital that they take in the clinician's office. That's a hint. One of the vitals that they take at the clinician's office. As soon as you go. They take it from me. Come on, they take your temperature, right? What do they take next?
Your oxygen?
Okay, pulse oximeter, oxygen. Then, an important one. Come on.
Oh, the blood pressure.
The blood pressure, yes, the blood pressure. So which means there are mechanoreceptors inside the carotid, right? That monitors the pressure of the blood. Baroreceptors, have you heard of them? So baroreceptor is nothing but a mechanoreceptor that senses the blood pressure, okay? And constantly gives information to the brain. And that's how sympathetic and parasympathetic evolves in our body. Got it? Interesting, right? So, and then thermoreceptive, changes in temperature, nociceptive. Just want to bring this term. What does nociceptive mean? Pain receptors. Okay, so any damaged tissue, right, injured or damage causes pain receptors to get activated. But anything that is strong from these can also activate pain. Yes or no? Too much pressure, too much of any other sensory information can also activate pain. And that's the unique thing about pain receptors. Okay? Dermatomes, just to give a clinical connection here, the dermatome is an area of skin supplied by sensory neurons that arise from a spinal nerve ganglion. So because of the different nerves, right? Anatomy, different components of different nerves, like cervical, thoracic, all of these, we can kind of gauge where the dermatome is, especially when a virus such as varicella zoster affects the body. All right, so that we know what nerves are getting affected and how they can be treated. So this is one of the things that follows the dermatome is the shingles. So the rash occurs in the dermatome of the infected nerve cell. And then we can take a closer look at how to treat that particular area in the body. Okay, again, this is for cord lesion purposes. Pain by itself is a lot of information, but just to give you some of the things that can go with pain is actually a good thing. Protective mechanism typically occurs when the tissue is damaged. Can also cause, typically, what does the body tell you? Hey, that there's pain, try to remove the painful stimulus, right? So that's the indication. But sometimes what we do, we just ignore pain, move on with our lives, right? That's a different story. The two types of pain, fast versus slow. So fast, again, there are different areas of the body that can get activated too, but just to keep it simple, fast kind of sharp pain, knife cut, needle poke, burn, type A delta fibers. And this is more superficial, right? Slow pain is like deeper tissues, and then kind of achy kind of pain, and we have type C. This is also where I just want to bring it in. It may not be on the study guide, like it's a requirement, but bradykinin, if you come across this particular molecule, this is associated with slow pain. Okay, just kind of an FYI if you need to, if you come across this in research articles. Okay. Ischemia causes pain. This is something that I want you to understand in terms of, these are important terms. So ischemia versus hypoxia. Ischemia is insufficient blood flow to provide adequate oxygenation. And hypoxia, because of reduced blood flow, what happened? The tissue is suffering from reduced level of oxygenation. All right, so basically they're deprived of what? Oxygen. If they're deprived of oxygen, right? And we know why ATP is crucial for many processes, right? And that causes, so basically the tissue starts dying without oxygen, without ATP. It can't control the electrolyte imbalance occurs. There's a lot of processes come to a halt. It basically causes tremendous loss to the body, resulting in pain. Okay, so again, arterial occlusion of limb. So this is kind of an experiment. So if you occlude, meaning if you kind of prevent the arterial blood supply, the pain starts within 50 to 20 seconds. That's how much the tissue is damaged because of loss of oxygen. So cause of ischemic pain, typically, these are some of the things that can prompt causing ischemia: bradykinin, proteolytic enzymes, lactic acid buildup. So this one, can anyone, biochemistry, quick tie up here, lactic acid? Okay, I will start.
Occurs like
Yes,
when
go ahead,
glycolysis
please.
is overactivated, like in the muscle.
I remember studying this all over the entire semester. Pyruvic acid, glycolytic intermediates. I remember, never forget. Glycolysis to pyruvic acid, then without oxygen, it becomes lactic acid. Yes or no? This is taken to the liver to process it. All right. So this lactic acid buildup means the body is deprived of oxygen. And another clinical connection here that I can make is septic shock. How many of you have heard of sepsis? Right. So sepsis is one of the, infection that can happen. That can also, how the clinicians assess is when the lactate levels in the blood build up. That can be an indicator for septic shock, which means that the body is deprived of oxygen. So it's not getting enough oxygen. So a lot of these things, I just want to kind of expose you to these things in case you are going into your clinical profession. Referred pain, how many of you are familiar with this concept? Somebody with gallbladder, they say they have some shoulder issues, or left arm pain can be associated with heart attack, right? That's a very popular one, correct? Right, so localized to dermatome of neurological origin, heart localized to neck, left shoulder, arm, stomach. So these are different things that you can see here. So what they're trying to say here, you see this overlap of sensory information going to CNS. The CNS is getting confused whether it's coming from the internal visceral organ or the actual somatic area. And that is why it is kind of radiating. So when these referred pain, they have come to a lot of quick diagnosis, right? Something that can be missed. So understanding referred pain can lead to osteodiagnosis that might otherwise be, especially with internal organs, correct? Gallbladder, liver, anything that's internal, these referred pain. And of course, if somebody has a shoulder injury because they fell, obviously it is going to get innervated with pain, but this is a venue that can be used for diagnostic purposes. Got it? Referred pain. Any questions? Yes. The nervous system, these special senses. How many of you are familiar with a little bit of anatomy of the eyes and the ear? So this is predominantly the eye, the ear, and taste. So it is a little bit of expectation where you would know the overall anatomy of the eye, right? So what is the what are these anatomical structures helping us with? So what is the stimulus? So the physiology here is what? The stimulus is the light rays. Yes or no? Right? From an object. So physiology is to convert this to what?
An impulse.
Exactly, impulse. Very good. Action potential on what nerve that takes it to the brain? Anyone?
The optic nerve.
Very good. So that's the job of the body, is to convert the light rays, correct? Guide it. So what inside the eye that basically receives the light rays? There should be something in the eye, correct? That does the action of, yes, Mala?
I think it's the retina.
Very good, retina. There you go. What do they have? Sorry,
They have
you
these
keep
like,
answering, I keep asking.
no, it's okay. I think I remember the rods and cones.
Very
The
good.
rods taking the white,
Yep.
the black and dark colors,
Yep.
and then
So
cones and-
retina, very good, very good. Retina has the photoreceptors. Remember, what is the light rays receptors? The light is looking for a reception, somebody to receive, warm welcome, right? We need to receive the light rays. So the receptors are found in the retina, okay? So optic nerve, the retina, you see that? Okay, so this is where rods and cones are there. So what are the job? What are the job of all the other structures? So you have the pupil, the opening, right? Light rays enter, correct? It is received. You have the transparent cornea. Then the cornea goes into this chamber called the anterior chamber aqueous humor, watery fluid. Okay, that provides nourishment to this lens. Nobody can beat our lens. You give me a lot of different iPhone lenses, but still our lens is the best. So we have the lens that is avascular. What does avascular mean? It does not have blood supply. So the nourishment comes from this aqueous humor. Humor means fluid, right? So which means if the lens uses up the fluid, the waste goes back into the anterior chamber, yes or no? Right? You use the fluid, nourish it, and then it dumps the waste. So the aqueous humor is constantly flowing in and out, in and out, in and out, yes or no? Right? So that's the beauty of that, which means if there is production issue or if there's drainage issue, what can happen to the anterior chamber of my eye? It's watery liquid, right? If there is overproduction or drainage problem, it could swelling. You can increase the pressure in the anterior chamber, causing pressure to the retina. Yes or no? You can put pressure on the optic nerve. What is that condition called? Increased intraocular pressure, causing damage to optic nerve. Got it? So that's how important aqueous humor is. Okay. So it is important for the body to maintain an optimum range of intraocular pressure with a constant flow of aqueous humor. Beautiful, right? So we have the eye and we have all of these amazing things. So the lens helps focus the light energy, correct? This is where physics comes in. We have a blend of biochemistry, physics, imagination, wow, anatomy, everything pours in, okay? So we have the light rays that needs the focus of fovea centralis. And then the retina has the rods and cones. Of course, this has rich blood supply coming from choroid. And then you have the opaque one, that's the sclera. So sclera is the opaque layer that prevents the light to go back. Yes or no? You have to capture it, process it, and send it to the brain. Correct? So that's why cornea is transparent. Sclera, the whites of the eye, is trans or opaque, right? Completely not letting the light rays to pass through. So these are all amazing anatomical structures that help guide the light rays to where? To the photoreceptors for processing. Got it? Okay. And then we have the vitreous humor. So vitreous humor maintains the shape of the eye. That's the fluid that's retained in the posterior chamber. Okay. So one of the things that we need to focus on is so glaucoma is one condition. Okay. The vision defects. So to understand this, we should know that you see this lens, they are held by, I want you to highlight ciliary muscle and then suspensory ligaments or zonular fibers. This really helps understand the defects, guys. Okay. Again, as simple as I can make, to see close by objects. To see close by objects, what do we do? The ciliary muscle contracts and suspensory ligaments, I'm just going to use abbreviation, suspensory ligaments relax. So this helps the lens to become more round. In physics terms, this is convex. Are you guys with me? All right. So if I see something very close, this is what happens to the shape of my lens. It becomes more curved or round. So if I'm seeing a distant object, the rays are going to be parallel, yes or no? Right? Distant object. So the lens will actually, to accommodate a distant object, it will become more flat, right? Okay. So ciliary muscles will, what happens to view distant objects? The opposite, correct? Ciliary body relaxes, suspensory ligaments contract, the lens becomes more, come on, distant object, the lens becomes more flat or concave. Are you guys with me? Yes? Everybody? So close objects, distant objects. So with this, let's look at this. So emmetropia. Emmetropia means normal vision. What does normal vision means? If I bring something close, it becomes round. If I take it far, it becomes flat. No issues, correct? That's emmetropic normal vision. What about this one? Myopia. Myopia is nearsightedness. This is one condition where we name the condition when it's actually the correct one. Able to see nearby objects clearly. Again, please remember one, the other one is opposite. Okay? Myopia is nearsightedness, able to see nearby objects clearly, which means where is the trouble? The trouble here is myopic eyes have difficulty focusing on
Far objects.
far away objects, distant objects, which means what are their lens not happening? They are not happening or they are not becoming
Converged. Like
flat or concave, right? Okay, so this myopia is trouble with distant objects. Got it? So what is the prescription? To prescribe them with? Does it make sense? The concave lens corrects nearsightedness. But if you don't know what nearsightedness means, then this understanding becomes a problem. Got it? So nearsightedness means you are able to see close by objects clearly, but unable to see distant objects. Got it? So if you know this, what would this be? The opposite, correct? Hyperopia is farsightedness, which means they don't have difficulty looking far away objects, but they have problem close by objects. So convex lens corrects farsightedness. Everybody good so far? Yes. Then astigmatism is problem both ways. So there's like uneven lens. So we just have to make an uneven lens to correct it. So if there is a bulge at the top, make sure that you try to even it out when you prescribe. Okay, so astigmatism is a kind of a combination of both. So they can't focus and they try to make it up with uneven. So far so good? Correction? Okay. So why to understand this helps. The mechanism. Okay. This intraocular pressure, IOP. So the range, so the rate of production of aqueous humor has to be constant, yes or no? We don't want too much or too less of it. Correct? We talked about glaucoma. Right? So typically the level of pressure, so there is something called canal of Sklen. What happens? This is the canal that helps drain it, correct? So if there is a problem with this one, so if there is, so this is blocked, let's say there is blockage. What is it going to cause? Do you think the aqueous humor is going to drain well? If there's a blockage here. So this is basically helps drain aqueous humor. We want to make sure that there is no blockage. So if there is a blockage, then it is going to cause high resistance. Yes or no? To the flow of aqueous humor. If there is high resistance, what happens to IOP, intraocular pressure, is going to be high. Eventually, it can cause glaucoma if left untreated. Yes, intraocular pressure, okay. And I'm touching on some of the main things here. So anatomy of the human here, how does this help? Again, what is the stimulus? Very similar to the eye. Our job for the ear is to convert. What is the stimulus?
Sound.
Very good. Sound waves to be converted to action potential, correct? And this is the vestibulocochlear nerve. Okay, that goes to the brain. Got it? So which means what is the sound wave looking for? Hearing receptors, correct? Right? Every time I say receptor potential, graded potential, those all things should come to your mind. Okay, so that doesn't change. So what are these doing? These anatomical structures doing? The pinna, the auditory canal. So all of these are what? They are trying to guide, yes or no? Or conduct the sound waves, right? They're trying to get the waves go into the ear. They're providing kind of a canal. So guide, conduct sound waves, and what happens? The tympanic membrane. Who can tell me a simple term for this? It's normally used.
The eardrum.
Very good. The eardrum, right? So the eardrum gets vibrated, vibration occurs, and that causes the middle ear cavity, which has the tiniest bones, malleus, incus, and stapes. Those are the tiniest bones in the body. They are actually found in the middle ear. That helps with kind of amplification, sound waves, and then they go into the inner ear. So inner ear is actually filled with fluid, surprisingly. The fluid is contained in the chamber, okay? And then cochlea, then you have the vestibule apparatus. So there are two things that are found inside the inner ear. So this helps with sensing balance. And this helps sense hearing. Okay, so again, these anatomical structures are helping to understand the nature of how things are processed in the body. Okay, are we good? The external auditory canal that takes it, middle ear, inner ear, cochlea. So now we are going to see a little bit of this and a little bit of this. Okay, just to see the end point. So we are in the cochlea, which means we are inside the inner ear. With me so far? Yes. So remember, the inner ear, up until the inner ear, what is the role of outer and middle ear? The role of outer and middle ear is to, like, one of the major roles is conduction of what? Sound waves. Correct? Okay. But where are the receptors found? Inside the inner ear. So inner ear has the receptors, which means this is where neural activity is going to happen. Yes or no? Got it? So if there is a damage to cochlea, what activity is impaired? The neural activity is impaired. Yes or no? Right? If there is a damage to somebody just poked something in the eardrum, what activity is impaired? Conduction is impaired. Right? So that is why we have two types of deafness. Conduction deafness, if there's a problem with the middle and the outer ear. Sensory neural deafness, if it is a problem with the inner ear. Boom, boom, sound all the time, Bose music. I'm not against any company, but a lot of music all the time can damage those tiny, tiny receptors inside the cochlea, causing sensory neural deafness. Make sense? To relate that, okay? So cochlea, again, we love anatomy. We are just going to focus on a few things here. There's a lot of anatomy here and everywhere. As a cochlea, I want you to go all the way to organ of Corey, right? So I'm just bypassing a lot of anatomical structures here. So that's one of the things that we need to do because our focus is still on physiology, right? Helps, okay? So organ of Corey has what? Hair cells. How do I relate hair cells? They are the receptor cells. Got it? What do you think these receptor cells do? Upon activation, they are talking to what nerve? Vestibulocochlear nerve. The cochlear nerve is what? The component of that will be activated, which means there will be action potential, and that will go to the brain. Yes or no? Right? So that's the physiology portion. Okay? So you see these organ of Cordy? Beautiful. You see those hair cells sticking out? That's the hair cell. So those are very delicate, guys. They can be damaged easily with a lot of sound. Are we clear? Yes? So inner ear, cochlea, organ of Cordy, hair cells, they talk to vestibulocochlear nerve. That will talk to the brain. Brain, we have hearing, temporal lobe. Does that ring a bell? That's where the hearing is processed. Okay. Moving forward, so I'm just going to point out they could be hair cells can have another different set of name called stereocilia. So hair cells, this is an important one. What I just told you. Hair cells are the receptor organs that generate APs. What does AP mean? Action, potential in response to sound vibration. That's all you need to know for the hearing. We're making it simple here. Yes? Yes, no? Okay. So see those cute little ones? Nice. What are they attached to? Cochlear nerve. That's part of the cranial nerve 8, which is a combination of vestibular and cochlea. Okay. So far, so good. The vestibular system, remember, what does this do? This is also in the inner ear. But this is sensing balance. There's a difference. So inner ear has two systems. So vestibular system, this vestibular apparatus senses balance and equilibrium along with other proprioceptors. So important here is we have the vestibule that has these structures that help with linear. What does linear mean? So for example, look at me. If I'm just doing this linear one way and just looking at it static and I'm not moving, right? So this is linear. So linear means static. Whereas look at me, if I do this, what am I doing? I am rotating, yes or no? I'm going in X, Y, and Z plane, yes or no? Right? X, Y, Z. You see that X, Y, Z? Amazing, right? So that's semicircular canals. So semicircular canals are rotational, or we call it dynamic. Do you think the body needs to know whether it's static or dynamic, guys? It needs to know, right? Whether that's when vertigo, all of those things are becoming important, right? So when there's problems. So again, knowing these, this is where anatomy comes in, knowing the structure. So if I say ampulla and cupula, what's the quick connection? Remember, these also have receptors, correct? These have, all of these structures will have receptors that will sense what? Balance. And these receptors are amazingly called hair cells. But the hair cells here are found where? In the vestibular system. If the hair cells are found in the organ of Cordy, what do these hair cells sense? Here. You see the difference? There are hair cells inside the organ of Cordy, but they are responsible for hearing. If I have hair cells inside these structures, they are responsible for sensing balance. Clear? Christy, Julianne, Trevor, Malak, at least from the Rom. Am I making sense so far? Yes? Okay. Again, as much as we cannot completely separate anatomy from physiology, guys, I hope you understand they're kind of intertwined like with each other. We need to know what it is to understand its functionality, right? I can't just say, hey, there's something called this, and that's the function. Does that make sense? You have to know what it is to understand its function. So there is a little bit of anatomy that goes with physiology all the time. So please be patient. And if you need me to clarify anything, please let me know. Okay, so the goal here is, yes, there are terms that we need to memorize, and that's where we need to put our effort on. So if I were you, I would take a blank sheet of paper or PowerPoint and say inner ear, organ of Cordy, hair cells, or inner ear. I would start with cochlea, organ of Cordy, hair cells, and then the nerve. Then I would have inner ear, vestibular system. Then I will divide that into vestibule, semicircular canals. What's under vestibule? Macula, right? Utricle and sacula. That goes under vestibule for static. Again, I'm still in the inner ear, semicircular canals, ampulla, cupula, dynamic. All have hair cells that talk to the nerve. That's it. It's just oversimplified, putting everything down so that it's easier to retain. Last but not the least, taste. So taste is very important, yes or no, right? That helps us ingest the food that we like and also helps with the generation of ATP, correct? Ultimately, our goal is to make sure that we are able to taste the food, and that helps with a lot of other things as well. So we have salty, sour, sweet, and umami. How many of you are familiar with MSG? Right, MSG comes under umami. Then bitter. Okay, so the great thing about this is we wouldn't have thought about how these are processed. So this is just to bring in the perspective of channels. salty food, rich in sodium, yes or no? Sodium is a cation. What do we know? Depolarization, right? So now it makes it easier to comprehend. Sour, acidic, H plus. H plus is a cation, right? Depolarization, yes or no? Pretty cool, right? So how the processing of taste occurs. Then sweet and umami. It is sugar or MSG, that the glutamate, binds to. This is where GPCR comes in, right? G proteins. Now, what does it do? It closes K plus. Make my day here. What do I put? Closing K plus.
So it'll depolarize.
Got it? Remember, K plus is stay put inside. K plus cation is inside, so it is depolarization. Yes or no? Okay, so that's why closing what ion channel is important. Opening what ion channel is important. Bitter, it activates G protein. It brings in calcium, which is cation, into the cell. We know calcium is high outside, coming in causes depolarization. Yes or no? Got
I
it?
have a question.
Yes.
Are these taste receptors only on the tongue?
Yes. So they are basically predominantly on the tongue. Yeah.
So like if you eat something sweet, it doesn't have to do with anything like depolarizing in the body. It's just on the tongue.
So they are kind of localized, and that's why they're categorized as special senses. So they are
Oh, okay.
localized. Obviously, it is going to go into the digestive tract, right? So that's also where you would take in sodium ions. The sodium ions are still going to be, so if you end up, or if I end up eating too much salty food, of course, the depolarization, it's going to activate the receptors, but the sodium is also going to go into my GI tract, and then it goes into the bloodstream, and that can also cause hypernatremia, like higher sodium levels in the plasma, and that will have effect on osmosis, you name it. So
Right.
the taste-wise, it's local, but it can still have a systemic effect on the body. Does that make sense?
Yeah, for sure.
Yeah, perfect. Thank you so much. I still have 3 questions. You still have the patience to finish those questions. Just go with the chapter. Yeah. Okay. Let's do it. Thank you so much. I think that's now. Okay, so finally we have the olfaction. So olfaction helps basically smell enhances taste, right? So I just want to point that out. Again, you have those receptors that help with olfaction and then goes into the olfactory nerve. So this is pretty straightforward, just to get exposed to these topics. We will do some review questions and then end our lecture for today, okay?